So today, Dr. Wang will speak on antibody glycosylation and site-specific ADCs. Dr. Wang, we're delighted to have you with us, so please take it away.
Thank you for your very nice introduction. I also would like to thank CBA for the opportunity for talking about our recent work. So today, let me show the slides first. So today, I would like to focus the discussion on glycoside-specific ADCs. Okay, just a second. Can you see the?
Yes,
the
we
lighter
can see your
pointer?
slides.
Okay, I just maybe just use this. Okay, so as you know, all the antibodies have sugars attached to it. For example, for typical IgG type antibodies, you will have conserved N-glycan as asparagine 297. This is highly conserved, so all antibodies will have it recombinant or natural antibodies. The sugars at this position, the FC, we call the FC glycans, is a typical complex type sugar. Cannot move it. Okay, so basically this is the symbols, for some in glycobody field may know it very well. But so you have two gluconate, we call it N-acetylglucosamine, and three mannose. So this is a core structure there. Outside of the core, you have decoration like a fucosylation called fucose, and galactosylation, cellulation. So all this addition of of the sugars can have a profound impact on antibody effect functions. So control glycosylation is fundamentally important for developing better or more effective antibody-based therapeutics. So today, actually, I will not talk about our work on the glycoengineering. Basically, I just give you one slide talking about how we can control the glycosylation as the Fc domain. So as you know, there are different methods. Like you can do metabolic glycoengineering, you can do biosynthetic pathway engineering to control the glycosylation. But that can have a lot of limitations because that will be limited by the biosynthetic pathway. So what in over the years, what we have developed is a so-called chemoenzymatic glycan FC glycan remodeling method that will allow you to actually control the glycosylation by introducing different types of sugars at the FC domain. Okay, so the method actually has two steps. The first step is to use an endoglycosidase to remove the heterogeneous sugar in the recombinant antibody by cleaving the glycosidic bond between the two N-acetylglucosamine, so you end up with a key intermediate in which the first sugar still attached to the glycosyl site. Then we develop the mutant enzyme based on the endoglycosidase structure. We do site direct mutation at a conserved residue that is critical for the hydrolysis. So once you do the mutation, you will eliminate the hydrolytic activity. However, if we provide with a high energy species like a sugar oxazoline as a transition state mimic, then the mutant enzyme can still take it for adding the sugar back to the original glycogen site. So by using the two steps, you will be able to remodel the FC glycan to construct a library of so-called homogeneous glycoforms of antibodies. So over the years, we developed different mutants and will be able to make a library of glycoforms with different sugars on it, and we have done some functional studies to identify the optimal glycoform for enhanced therapeutic efficacy. So that is one of the line for the research in the lab. But today, the question I would like to ask is whether the Fc glycan can also provide the handle for site-specific antibody conjugation. And the answer is yes, because there's so many people who have done, the Fc glycan modification to introduce payloads on the Fc site. So Dr. Chindi has given a very nice introduction of the ADC field. I don't need to repeat it right now. So basically, for a good ADC or effective ADC, you have to have the right choice of the target and the antibody, then the selection of the payload, the linker, and of course, the conjugation chemistry that will put things together, right? So that, of course, there are many excellent methods that have been developed in the field. The original, of course, random conjugation, as Dr. Lee has mentioned, have a lot of problems. And then the disulfide bound reduction conjugation is one of the methods for most of the ADCs recently approved by FDA. Then you can engineer the protein to introduce the cysteine for conjugation. You can introduce an unnatural amino acid. You can use proximity-enabled chemistry to do the chemoselective conjugation or introducing fusion tags then with subsequent conjugation. And also in the medical coupling like a MTG. So today I will focus on the Fc glycan as a handle for site-specific conjugation. Okay, so as I mentioned, the antibody has an Fc glycan, and it's a heterogeneous, complex bi-attendant complex of sugar, right? But all the side sugars can be subjected to modification to introduce the tag and do subsequent click chemistry for conjugation, right? People modify the fructose and galactose and even introduce the salicylic acid with the tag, and then you can do click chemistry. Okay, so there are two, I think, notable examples in the field. Like GSKN003 by Alpha Map, So this is an anti-HER2 bispecific ADC, and now it's in phase 3 clinical trials for breast cancer, ovarian cancer, and other cancers. So for this ADC, they use the chemistry again is modification of the Fc glycan. So for that, you have to remove the outside galactosalic acid for the antibody, and then you introduce an azide-tagged galactose derivatives called GalNAcs by galactotransferase. Then you do click chemistry, infuse the payload. And another example is the IBI343 by Innovent Biologics. So this is an anti-Claudin 18 antibody, and now in phase 3 clinical trials for pancreatic cancer. So this uses actually the Synaffix technology by engineering the Fc glycan. So it's to use the endo enzyme to remove all the N-glycan, leave the first sugar on, then use a galactotransferase mutant to introduce GalNAcs with azide tag, then you can click the payload on the antibody. Okay, so these are two, I think, very promising antibody drug conjugates, for the treatment of cancer. It's a kind of proof of concept for the Fc glycoside specific antibody. Okay, for our own work in this field, basically, we use the technology we developed to do the Fc glycan remodeling. The first generation method is to modify the salicylic acid by introducing azide tag, because here you can use two steps, right? You can use a Y-type enzyme to remove the Fc glycan. Then you use the mutant to transfer the enzyme-detected sugar back. So now you have enzyme-detected antibody, and click chemistry you provide the ADC. The advantage of this method is that it doesn't matter how you produce the antibody. You can use yeast cell, insect cell, and mammalian cell because you don't care about the glycan. So long as you have the glycan, you can remove it first and then put the tag on, right? So this technology was licensed by to Daiichi Sankyo a few years back, and currently they have ADCDS 9606 in clinical trials for advanced solid tumor. So they have worked out the larger like a scale preparation of the complex drug with the tag, but still the drawback is the tedious procedures for the preparation of the natural N-glycan by isolation from chicken egg yolks. Okay, so the question is like, can we develop a simple method just to simplify the donor substrate? So for that purpose, actually, we synthesize a number of simple oligosaccharide down to disaccharide by introducing the azide into the disaccharide oxazoline. And surprisingly, we found like a Y type of endoglycosyl, like the NS2, can do the hydrolysis of natural FC glycan, but at the same time, it can take the azide modified disaccharide to transfer to the antibody. So you can combine the two steps and you don't need to have two enzymes. So Y-type enzyme will be sufficient to do the simultaneous deglycosylation and reglycosylation. So with the azide, you can do click chemistry to reduce the payload. By controlling the number of azide on the disaccharide, you can have the variation of the di numbers. Okay. And also we can preload the drug on the disaccharide, and then you use the Y-type endoglycosis to do the remodeling of the FC glycan. So a single step, single enzyme, one pot, you can get the ADC. Okay, and you can use different disaccharide that of course have different activities. And so we did the cell-based A cells, as you see, is potent ADC. And we did some characterization of the ADC synthesized. And we found in general, with the type 2 to 6, you can have like a, quite a high stability and maintain FC ion binding. So, and reduce FC gamma 3 bindings that may reduce some of target toxicity. And in the animal models, you can see the efficacy, right? By a single injection, you can shrink the tumor and at the end, the tumor will disappear. Okay. So we also apply this method for, antibody ligand conjugate for lysosome targeting chimera design. So here you can basically use the FC glycan remodeling method to introduce -6-phosphate oligosaccharide in a single step. And we have shown that can actually lead to the degradation of the membrane-associated target. And also we extend this to the trigalNAC ligand for a seroglycoprotein receptor for the degradation of extracellular proteins. Here we use PCKS9 as an example. We show actually the conjugate can efficiently reduce the level of PCSK9 in cell-based studies. So in conclusion, we accept the chemoenzymatic glycan remodeling method for site-specific antibody-drug conjugates, and the single enzyme one-pot approach provides a general platform for producing site-specific antibody-drug and antibody-ligand conjugate for further therapeutic development. Okay. So with this, I would like to acknowledge people who actually made the intellectual contribution to the initial stage of the work, and glycan etching moves on with the technology for further ADC development. Thank you.